All right, it's my pleasure to introduce Nithin. He was from India and is an accomplished fisheries scientist with an extensive background in aquaculture and aquatic animal health management. He currently serves as an Oak Ridge Institute for Science and Education postdoctoral fellow in fish functional genomics at the USDA ARS facility in Auburn. Take it away.
Hello. Yes, sir. Thank you very much, sir. Myself, Nithin MS, and I'm working under the guidance of Dr. Jason Abernathy in USDA ARS Aquatic Animal Health Research Unit. Today, I will be presenting the topic on novel lytic bacteriophage AHFM11 as an effective therapeutic agent against hypervirulent Aeromonas hydrophila. As we know that aquaculture is growing, and similarly, the pathogen as well as disease occurrence, and it caused an economic loss of 6.5 billion USDA. Among the bacterial diseases, Aeromonas is one of the disease-causing agent which causes motile Aeromonas septicemia as well as furunculosis. And in order to, because already Yashutor has talked about hypervirulent Aeromonas hydrophila, I am not going to talk about that part. So coming to treatment aspects, here the approved drug that is tetracycline is used in aquaculture, but due to the occurrence of antimicrobial resistance in aquaculture, one study conducted by Reverter et al. Showed that global multiple antibiotic resistance index in case of aquaculture derived bacteria was higher than 0.2, which indicates the presence of multi-drug resistance bacteria. The most of the orange color indicates the presence of multi-drug resistance bacteria in the environment. So as an alternative, we can go for prebiotics, probiotics, or vaccines, or we can go look for the presence of the bacteriophages. That is, bacteriophages is nothing but the viruses which specifically act on a particular bacteria. So here, phage therapy can be used in different approaches, such as conventional phage therapy, where we can use monophage therapy, as well as polyphage therapy, where in case of monophage, only one phage is used, as well as in case of polyphage, where we can use different phages acting on particular species of bacteria. And also immunophage antibiotic synergy can be used, where we can use combined effect of three phage antibiotic as well as immune vaccinated vaccine developed in a combination. And also we can use phage-derived enzymes such as endolysins and also biongenophages. Based on this, we created a hypothesis that whether these phages can be used for treating the hypervirulent Aeromonas hydrophila in aquaculture systems. So based on that, my objective is set. First of all, we have to isolate the Aeromonas lytic phages from the aquatic system, and then we have characterized them genomically. And the second objective is to use in vivo experiments with Aeromonas phage against hypervirulent Aeromonas hydrophila as an biocontrol measure. Coming to the isolation part of bacteriophages, we collected the samples, centrifuged it, and filtered it, and we did the spot test based on the Van et al. And we found that there is presence of the clear zone, then which indicates the presence of these phages. So this is the methodology what we have followed for isolating the phages and characterization of the phages. And I will not be talking about all of them, but I will be talking only few selected part. That is host range, one-step growth curve, absorption test, as well as genomic characterization and in vivo experiment. Coming to host range, we have selected 92 Aeromonas species and performed the spot test. And we have repeated this experiment 3 times by using a AHFM11 phage at a titer value of 10 to the power 6 to 7 plot forming unit. Main objective is to know how many bacteria it can lyse. Coming to one-step growth curve, the main objective over here is to determine the burst size. Burst size is nothing but the number of viral particles coming out of one particular bacterial cell. And whereas in case of adsorption test is done for determining the rate of adherence of viruses to the bacterial cell. And also we perform the electron microscopy in order to determine the structural morphology of the isolated phages. And also we did the whole genome sequencing by using Illumina pyrosequencing method, and also perform bioinformatic analysis and comparative genomic study in order to determine why these phages are better. Coming to the in vivo experimental setup, here we have designed three different method of approaches, that is injection method, immersion method, and feed method. In case of injection method, we have injected the phage right after the challenge. So we have challenged with the hypervirulent Aeromonas hydrophila at 1.5 into 10 power 6 colony forming unit per ml. And after 2 hours, we have injected the phage AHFM11 at 10 power 7 plaque forming unit. And we have checked for the survival for 15 days. And whereas in case of immersion, we have challenged it and immerse the phage in water for 30 minutes in case of a 10 power 7 plaque forming unit and observe the survival for 15 days. And whereas in case of feed, we have given phage impregnated feed group for 60 days. And after 16 days, we have challenged with the hypervirulent Aeromonas hydrophila and we recorded the survival. These two challenge group with phage as well as control group serve as the positive and negative control in our study. Coming to the results, we have isolated the hypervirulent Aeromonas hydrophila from koi fish, which is forming the beta hemolytic hemolysis in blood agar. And these are the spot tests which we performed, where we can able to clearly see that the phages are present, and we perform the phase titer, and we can the AHFM11 phase titer was 6.2 into 10 power 9, whereas here in case of host range analysis, we found out that out of 92 Aeromonas species, the phage can lies about 75 Aeromonas hydrophila, and hence this can be used for the therapeutic purposes as well as which are reported by several other authors. This is the electronic microscopy of HFM11, that is transmission electron microscopy. The first figure indicates the plaque assay, where you can see clear plaques at 1 to 2 millimeter. And also the second picture, you can able to see that icosahedral, that is electron microscopy, icosahedral head of phages with the neck as well as spikes and tail fibers. And this belongs to the family virus of myovirus and family of Straboviridae. Recently, International Committee on Taxonomy of Viruses classified, abolished the Myoviridae family and classified new group of families such as Straboviridae. That is also reported in Turner et al. 2023. Coming to adsorption and one-step growth curve, here we can able to see that the bacteriophages adsorb at a rate of 99.7, and no phages were available after 30 minutes, and hence it is have a high adsorption efficacy. And whereas in case of burst size determination, the latent period was 10 minutes and the burst size was about 378 plaque forming units of viruses per host cell. Coming to genomic characterization of the phages, we have isolated and extracted the DNA from that and sequenced it and found out that the AHFM11 has a genome size of 168,243 base pair with a GC content of 41.5%. And we have submitted it in NCBA with this accession number. And we have found a wide range of MCPs, endolysins, prophase systems in the genome. Based on that, we used the major capsid protein for characterizing the evolutionary to know about which group it highly belongs. And here there are 5 clades, that is nothing but group 1, which are having the Aeromonas salmonidophages, group 2 having Aeromonas hydrophilophages as well as other phages belonging to myovirus. Group 3 also is Aeromonas phages belonging to Autographivirinae family. And group 4 is nothing but Enterobacteriophages, phages belonging to Enterobacteriophage bacteria. And group 5 is where my phage belong to group 5 with other 2 phages, that is 50 Aeromonas hydrophila as well as 60 Aeromonas hydrophila, and belongs to second of 5 virus. And also in the genome of Aeromonas hydrophila phage, we found no antibiotic resistance genes as well as virulent gene. Coming to comparison, because we wanted to compare with the Aeromonas phage 60 as well as Aeromonas phage 50, because they are forming the same clade. And we found out that more than 85% of genome match between those groups. And we come to know that these 2 phages have been used in different studies, and they have developed a commercial product named Befador for treatment of Aeromonas hydrophila and Pseudomonas diseases in eel in Poland. And based on this, we thought to use it in phage therapy. In case of injection, we found out that 98.5% survival when we use it when compared to the control groups. Whereas in case of immersion group, we found out that 95.11% survival when compared to the control with challenge group. And in case of phage impregnated group, we found out that there are 93.11% survival when compared to the control with challenge group, and no mortality was observed in case of the control which we injected with the PBS. Overall, coming to the conclusion that phage therapy can be used in aquaculture to solve motile Aeromonas septicemia as well as antimicrobial resistance, and hence this can be applied to different projects such as biocontrol, food industry, and medical industry. And thank you very much.
Great, thank you. That was a great presentation and interesting approach.
Thank you, sir.
Folks, we have a couple minutes for questions. If you have them, feel free to drop them in the chat or unmute yourself. We have one here in the chat. Nithin, what do you believe would happen if you allowed the initial disease challenge to go beyond 2 days, say perhaps 1 week?
For challenge group,
I guess so. Dave, feel free to unmute yourself if you want to clarify the question.
Yeah, can you hear me?
Sure
yes, I can hear you.
The question is what would happen if you infected the fish and let them go for a week before you applied your therapy?
That experiment, we have done it in different approaches. So we have done it with 24, 48, and 72 hours. If we challenge after 72 hours, we found out that most of them will die of the infection.
So in practicality, how can a fish farmer apply this during an outbreak?
During an outbreak, first we have to really know that it is caused by Aeromonas hydrophila within 24 hours, then we can able to apply this.
Okay, any other questions out there? Okay. Thank you very much, Nithin.
Thank you, sir.